Today, I will be talking about mRNA processing. Per the central dogma, DNA is transcribed by RNA polymerase into pre-mRNA. There is also co-transcriptional processing of pre-mRNA into mature mRNA. Mature mRNA is then translated into proteins. However, I will be focusing on the co-transcriptional processing of pre-mRNA. During transcription, the 5' end of pre-mRNA is capped with 7-methylguanosine. The cap is also bound by cap-binding proteins to shield the cap from degradation by exonucleases. There is addition of a chain of adenine to the 3' end too. More so, an intron removal process or splicing of pre-mRNA occurs to form a mature mRNA. It is important to note that prior to polyadenylation, a series of events take place, and I will be talking about those events shortly. The core polyadenylation proteins consist of cleavage and polyadenylation specificity factor, cleavage stimulatory factor CSTF, cleavage factor 1, cleavage factor 2, and poly-A polymerase. Here we see that RNA polymerase 2 C-terminal domain serves as an assembly surface for the processing factors. CPSF and CSTF bind to the poly-A signal AAU, AAA, and U or GU-rich regions respectively, where binding of CPSF facilitates cleavage of pre-mRNA about 10 to 30 nucleotides downstream of its binding site. This is followed by degradation of the downstream 3' fragment. Upon cleavage by CPSF and degradation of the downstream 3' fragment, the enzyme polyadenylate polymerase comes in to synthesize a long chain of adenines to make a poly-A tail in what is called polyadenylation of mRNA tail. Additionally, a polyadenylate binding protein binds to the poly-A tail to prevent degradation by exonucleases. Thank you for your time and attention. I believe we have learned a lot about pre-mRNA processing.